Welcome back to the deep dive. Today we are focusing on an organism that's just 1 millimeter long, about the size of a comma on a page, but its impact on human biology has been, well, just astronomical.
It really has. We're talking about the tiny nematode Caenorhabditis elegans, or C. elegans for short.
And if you're trying to get up to speed on modern biomedical history, this is your shortcut. This tiny worm shares something like 13,000 genes with us.
Yeah, thousands of which are directly involved in human diseases. It's a classic scientific paradox. Minimal size, maximum insight.
So our mission today is to unpack not just the big discoveries, and we're talking 4 Nobel Prizes, but the unusual collaborative community and the shared infrastructure that made it all happen so fast.
And that's really the key to understanding this kind of research. The reason we study the worm is because evolution is, well, it's lazy in a good way.
Yeah.
It reuses things. So these ancient conserved biological processes, like how our cells develop or age, they're shared across huge evolutionary distances. You find a core mechanism in C. elegans, and you've basically found a blueprint for human biology.
And you often end up shining a light on parts of our own systems we never even knew were there.
Exactly.
Okay, so let's start at the beginning. The choice of the organism itself. We're going back to the early 1960s with Sydney Brenner.
Brenner was looking for an animal that was simple enough to tackle these huge questions, development, neuroscience, but with the new tools of molecular biology.
And this was a pretty radical move at the time, wasn't it?
Oh, a huge one. I mean, the reigning champion of genetics was the fruit fly, Drosophila. So Brenner was essentially betting his whole career on this tiny, barely visible roundworm.
But his logic was sound.
It was impeccable. C. elegans had a rapid life cycle, it was completely transparent, and it had a simple invariant anatomy.
Meaning every single worm looks exactly the same, cell for cell.
Precisely. Which made it perfect for genetic analysis. And that precision, it just immediately created this incredible culture of collaboration.
Long before the internet, right? They were already building this community resource.
Absolutely. The whole ethos was open science from the get-go. You have to mention the Worm Breeders Gazette. Bob Edgar started it in 1975. It was basically a newsletter where scientists shared everything, unpublished data, mutant strains, new methods, before any kind of formal peer review.
And that tradition of open data, which John Sulston and Bob Waterston really championed, didn't it set the stage for the Human Genome Project?
It really did. It set the precedent. And it's what allowed C. elegans to become the first animal to have its entire genome sequenced back in 1998. The achievements were just monumental. The full cell lineage, the complete nervous system wiring diagram, and then that whole genome.
And that brings us directly to the Nobels. Let's start with the 2002 Prize for Programmed Cell Death, shared by Brenner, Robert Horvitz, and John Sulston.
Right. And this is where you just have to appreciate the sheer difficulty of the work. John Sulston took on this heroic task of chasing every single cell division from a single fertilized egg all the way to the adult worm.
Manually.
Imagine sitting at a microscope, tracking all 1,090 somatic cells in a living animal. It's a feat that has never been replicated in any other animal.
And what did he find?
Something astonishing. He found that of those 1,090 cells, exactly 131 of them died.
Just died.
Yeah, naturally and predictably. It was a process of cellular self-destruction. We call it apoptosis. This wasn't damage. It was a feature baked into the developmental program.
So he knew the what. What about the how? The mechanism?
That's where Horvitz's work came in. Through genetic screens, he identified the molecules, the actual genes responsible. And what they found was this fundamental balance. You have 3 executioner genes that promote cell death, AGL1, SED4, and SED3.
Okay.
And then you have a single guardian gene, SED9, that blocks the process. It's like a switch that we all have.
And the connection to human health was immediate.
It was direct. The human version of that worm guardian gene, SED9, is a gene called BCL2. And if BCL2 is overexpressed, it stops cells from dying when they should.
Which leads to cancer.
Exactly. Specifically follicular lymphoma. So understanding this switch in a worm paved the way for drugs like venetoclax, which basically turn that death switch back on in cancer cells. It's a perfect line from basic worm science to life-saving medicine.
And the speed of all that research, it relies completely on the infrastructure. Which brings us to the physical library, the Caenorhabditis Genetics Center, the CGC.
Yeah, established in 1979. The CGC is the ultimate expression of that cooperative spirit. It's not just a collection, it's a global public utility. It makes the research more efficient, more rigorous, more reproducible.
Instead of every lab maintaining its own strains.
Right. Why should every lab dedicate staff to that? They just rely on one central, high-quality repository. And the scale is staggering. We're talking over 27,000 different cryopreserved strains.
Wow. And you can see the impact in the numbers. The Horvitz lab alone made 917 useful strains. The CGC has sent those out over 40,000 times.
It frees up researchers to just do the science. And it's also a critical safeguard. I mean, these stocks are frozen in liquid nitrogen. Some have been viable for over 40 years. And get this, they have an off-site ARC.
An ARC.
Yep. A complete frozen copy of the entire collection stored in Fort Collins, Colorado. So if there was a disaster at the main facility, the engines for future discoveries, as they call them, are safe, essentially forever.
That is just amazing foresight. So that physical collection was the foundation. But as the data exploded, especially after the genome was sequenced, the community needed a digital backbone.
The volume of information just demanded it.
Yeah.
And with almost 40% of the worm's genes having human equivalents, it was a massive organizational challenge. This all sets the stage for the next Nobel, the 2006 prize to Andrew Fire and Craig Mello for RNA interference.
RNAi, and this was another one of those completely unexpected findings, right?
Totally. What Fire and Mello found was that double-stranded RNA could act as this incredibly potent and specific trigger to silence a gene. They didn't just find a new lab tool. They uncovered this ancient, universal mechanism that cells use to regulate their own genes.
Or to fight off viruses.
Or to fight off viruses, exactly. And the translational jump was immediate. RNAi kicked off these huge, large-scale functional genomic screens in the worm. You could suddenly knock down every single gene one by one and see what it did.
And that workflow, that whole paradigm, it became the model for newer techniques like CRISPR screens, which are now standard everywhere.
And the path to the clinic was just as fast. Today, we have FDA-approved RNAi drugs like Onpattro that treat rare genetic disorders by just silencing the one bad gene. And it all started by injecting some double-stranded RNA into a transparent worm.
Incredible. So
Yeah.
to handle all this new genetic information, we get WormBase, which was established in 2000.
Yeah, WormBase is the source of truth. It's not just a data dump. It's the single place where all these different data types, gene sequences, protein interactions, reagents, are all translated into one standardized language.
And it feeds into the bigger databases, right?
It does. It feeds vetted data to huge resources like RefSec. What I find so compelling is the connectivity. It integrates seamlessly with the physical strains at the CGC, and it links published data with unpublished stuff. Even those old Worm Breeders Gazette articles.
Yeah.
You have decades of detail at your fingertips.
And it's part of a bigger alliance now, too.
It is a founding member of the Alliance of Genome Resources. So it's working with the mouse, fly, and yeast databases to standardize everything, which helps the entire field.
Okay, moving from the digital back to the visual, the 2008 Nobel Prize, Martin Chalfie, for the discovery and development of green fluorescent protein, GFP.
Right. And Chalfie himself credits C. elegans for the big aha moment. Because the worm is transparent its whole life, it was the perfect testbed. He just needed to know, could you put the gene for GFP into another animal and just see it light up?
And it worked.
It worked brilliantly. It confirmed GFP was this outstanding marker. You're essentially giving scientists a tiny headlamp to watch biology happen in real time.
In vivo, in a living animal. That's the key.
That was the revolution. Before GFP, this was so complicated, often destructive. After, you could trap pretty much anything. Protein localization, how HIV spreads between cells, how cancer metastasizes, all with these glowing tags that came directly from that first simple worm experiment.
So the worm gave us the living lens. Okay, so we have the strains from the CGC, the genetics from WormBase, which brings us to the third critical pillar, the anatomical context. And that means WormAtlas.
WormAtlas, yeah.
Yeah.
Started in 1998. Think of it like Netter's Atlas of Human Anatomy, but for the worm. It maps out the physical structure of both sexes, even specialized stages like the dower larva.
And a big part of its job is just archiving, right? Preserving this anatomical data.
Absolutely vital. It safeguards the intellectual heritage of electron microscopy, EM, data. We're talking about 11 shipping crates full of the original archival data from the 70s and 80s that map the entire nervous system.
And that's not just sitting in a basement somewhere.
Not at all. It's continuously being digitized, reevaluated by modern scientists, sparking new ideas about how nervous systems are built. And now they're expanding to do comparative anatomy with parasitic nematodes, the ones that cause human disease.
Which brings us to the most recent prize, the 2024 Nobel to Victor Ambros and Gary Rothkun for discovering microRNA.
Yet another discovery that was completely unexpected. It came from studying two genes, LIN4 and LET7, that controlled developmental timing in the worm.
And again, it's the worm's simplicity that made it possible. Its invariant cell lineage, its transparency. It made the mutant phenotypes just so obvious. You had cells that were temporally confused, like they were reliving a past stage of development.
It was striking. And then came the discovery that this mechanism was deeply conserved. The Let7 microRNA sequence is nearly identical across all bilaterian animals.
So everything from worms to us.
Everything. This showed that microRNAs were a fundamental way of regulating genes after they've been transcribed. They're like dimmer switches for genetic output.
And like RNAi, they quickly found a role in medicine.
As diagnostic and prognostic markers for all sorts of diseases.
So if you put it all together, what you're really seeing is this masterclass in cooperative science.
It really is. The CGC gives you the standardized physical material, the strains. WormBase gives you the standardized genomic knowledge, and WormAtlas provides that crucial anatomical context.
And that synergy makes the research incredibly rapid and economical. Foundational breakthroughs like understanding apoptosis or discovering RNAi probably happened years, maybe even decades sooner because of it.
It's a powerful demonstration. Curiosity-driven science in a simple animal has a direct line to human health, and even to other fields, like using RNAi to develop agricultural pest controls.
The legacy isn't just the papers. It's the infrastructure itself, the physical and digital assets, frozen and archived, just waiting for the next generation.
Exactly.
Which leaves us with a final thought for you to chew on. We mentioned that WormAtlas is safeguarding those original electron microscopy images, 11 crates of them from the 70s and 80s. Now, considering how new technologies, new ways of digitizing things can rekindle interest in old data, what unexpected discovery might be waiting right now in those decades-old crates, ready to be scanned and reevaluated by a new generation of scientists? That is what we mean when we talk about the profound value of safeguarding our scientific heritage.